Gastric cancer. It's a significant challenge, Fifth most common cancer globally, and sadly, the fourth leading cause of cancer death.
Yeah, it's a tough one. And a big reason it's so deadly is that it's often caught really late.
Exactly. When treatment options are more limited, even with surgery or chemo, the outcomes for advanced stages, well, they aren't great.
Which highlights this real urgent need for better tools, ways to find it earlier, track how it's responding, guide treatment better for each person.
So today we're doing a deep dive into something that holds a lot of promise here: liquid biopsy in gastric cancer. Now, if you're not totally familiar, the basic idea is getting molecular information about the tumor, but not from cutting out tissue. Instead, you use a body fluid sample, usually blood, but we'll see there are others too.
And our guide for this dive is this really comprehensive review paper. It pulls together findings from a whole bunch of different studies.
What's special about it?
Well, it's scope, mainly. It doesn't just zero in on, say, blood samples or just DNA fragments. It looks at various liquid sources and all sorts of molecules circulating tumor cells, different RNAs, DNA, even these tiny things called exosomes.
So it gives a really broad picture.
Exactly, a landscape view of the research. And importantly for our discussion, it organizes a lot of this using some really clear figures and tables, which we'll be leaning on.
All right, let's unpack this then. Our mission is to explore what liquid biopsy might offer for gastric cancer, drawing insights directly from this review, especially focusing on that data shown in the figures and tables.
We want to give you a clearer picture of the potential, the key findings, without getting totally bogged down in every scientific nuance.
Makes sense. So maybe start with why we even need liquid biopsy. How does it compare to what we do now?
Okay, so traditionally, diagnosis and understanding of the tumor relies on imaging scans and then a tissue biopsy, actually taking a piece of the tumor.
Right, the standard approach.
But there are limitations. By the time you can see a tumor on a scan, it's often already pretty big. We're talking maybe 1 to 5 billion cells. That's not early.
Yeah, that many.
Yeah. And the tissue biopsy itself, it's invasive, right? Needs a procedure. Plus, it only gives you a snapshot.
A snapshot.
Yeah, of one specific part of the tumor at one single point in time. And the problem is tumors aren't uniform. Different areas can have different genetic changes. That's tumor heterogeneity.
Ah, okay. So a single sample might miss important information.
It can, yeah.
And that's where liquid biopsy comes in, offering an advantage.
Precisely. First off, it's minimally invasive, often just a standard blood draw.
Much easier on the patient.
Much easier. And easier to repeat. Now, if you look at figure 1 in the review, it shows us samples from blood, yes, but also potentially saliva, urine, gastric juice, other fluids too.
Right, I see that diagram. Multiple potential sources.
And because it's easier to collect samples repeatedly, you can do dynamic monitoring, track changes over time, how the cancer evolves, how it responds to treatment.
So not just a single snapshot, more like a movie.
Kind of, yeah. And because the sample, like blood, circulates through the body, it might capture material shed from different tumor sites.
Yeah.
Giving you a more sort of whole body picture helps overcome that snapshot limitation of the tissue biopsy.
That makes sense. Capturing more of the tumor's complexity.
Exactly. And getting that kind of real-time, comprehensive molecular picture is, well, it's becoming really critical for precision medicine.
Yeah.
Tailoring treatments.
Okay, so we collect these fluids, blood, saliva, whatever. What exactly are we looking for in there? What holds the information?
Good question. The fluids contain stuff shed by the cancer cells or released by the surrounding environment. The review breaks it down neatly into 2 main categories: cellular and acellular components.
Cellular, like actual cells.
Right. The review mentions circulating tumor cells, CTCs. These are actual cancer cells that have broken off the tumor and are floating in the bloodstream.
Okay, escapees from the tumor.
Pretty much. And they also mention peripheral blood, mononuclear cells, PBMCs. These are immune cells in the blood.
Why are those important?
Well, CTCs are thought to be involved in metastasis, the spread of cancer. And PBMCs can tell us about the body's immune response to the cancer.
Yeah.
But the review does note a big challenge with CTCs. They're incredibly rare, hard to find and study.
Like finding a needle in a haystack, I imagine.
Something like that, yeah. Then you have these cellular stuff molecules just floating around.
Not cells, just molecules.
Exactly. Key ones are cell-free DNA, cfDNA. These are fragments of DNA released when cells die, normal cells and cancer cells.
Okay.
And the really important bit within that is circulating tumor DNA, ctDNA. That's the fraction of cfDNA that specifically comes from the tumor cells, carrying their mutations and alterations.
Got it. Tumor DNA floating in the blood.
Right. The review also covers non-coding RNAs, ncRNAs. These regulate genes but aren't turned into proteins. Then exosomes, tiny little packages released by cells. They can carry and protect other molecules like RNA or proteins, making them more stable in fluids.
Like little molecular mailbags.
Yeah, that's a decent analogy. And finally, proteins themselves, which have, been used as biomarkers for a long time.
Okay, so quite a range of things to look for. The review has another visual, figure 2, that seems to tie this together.
Yes, figure 2 is really helpful. It basically maps these different biosources, blood, non-blood fluids, and the components we just discussed, to their potential clinical uses.
Ah, I see. It connects the dots.
Right. It shows how researchers are hoping to use CTCs, ctDNA, ncRNAs, exosomes, and so on for things like early diagnosis, checking for minimal residual disease after surgery, monitoring for recurrence, predicting prognosis, and guiding treatment choices. It's a nice flowchart of the goals.
Okay, that clarifies the what and why. Let's dive into the how well does it work part. You mentioned blood as the most studied source.
By far, it's just the easiest to get right, a standard blood draw. So analyzing blood gives us the best shot right now at getting that dynamic, system-wide molecular picture.
So let's start with the findings from blood. Early diagnosis seems like a huge goal. What does Table 1 in the review tell us about that?
Okay, yeah, Table 1 summarizes a lot of studies on blood biomarkers for early GC diagnosis. And the takeaway, it's still pretty challenging.
What is that?
The main issue is sensitivity. When a tumor is really small, truly early stage, the amount of stuff it sheds into the blood, like ctDNA, is often incredibly low, just very hard to detect reliably.
Makes sense. So what approaches are they trying based on the table?
Well, for cfDNA, instead of just quantity, they're looking for specific tumor patterns, like DNA methylation. Table 1 mentions things like the CancerSeq test. That's a multi-marker test. It showed decent results overall for several cancers, but the sensitivity specifically for gastric cancer was lower, maybe around 40% in the studies cited there.
Not ideal for screening yet.
No. But other approaches using methylation panels, like one called EpiPan-GI-Dix mentioned in the table, showed better potential for GC, with an AUC around 0.900. Remember, AUC measures accuracy, 1 is perfect. The table also lists specific methylated genes being looked at, like RUNX3, SFRP2, with varying results.
What about those non-coding RNAs you mentioned? Table 1 seems to have a lot on those, especially miRNAs and things in exosomes.
Yes, that's a really promising area highlighted in the table. The review notes ncRNAs can be quite stable, especially when they're packaged inside exosomes. And the data in Table 1 reflects this. You see studies with specific miRNA panels reporting quite high sensitivity and specificity.
Like?
Well, one 3-miRNA panel cited shows numbers over 85% for both sensitivity and specificity. Another 4-miRNA panel reported numbers almost hitting 100%. And exosomal long non-coding RNAs like LNCU-EGC1 also show really high-performance metrics in some studies listed there.
Even CPCs show up in Table 1 for diagnosis, despite being rare.
They do. One study cited by Kang et al. Reported decent sensitivity and specificity, around 85–90%, with an AUC over 0.9. So multiple angles are being pursued for early detection in blood, though it's clearly still under development.
Okay, moving past diagnosis, what about after surgery? Monitoring for recurrence. Table 2 focuses on this, right? Minimal residual disease, MRD.
Exactly. MRD is crucial. After surgery, you want to know if any tiny bits of cancer were left behind. Traditional follow-up, like scans, often can't pick up these microscopic amounts until they've grown quite a bit.
And Table 2 suggests liquid biopsy, particularly ctDNA, could be better.
That's the hope. And the data in the table is encouraging. It shows that detecting ctDNA in the blood after surgery is strongly linked to a higher risk of the cancer coming back and generally poorer outcomes. Often, ctDNA positivity shows up months before recurrence is visible on scans.
So it's like an early warning system.
Potentially, yes. The review discusses different ways to look for ctDNA for MRD. Tumor-informed approaches are often preferred now.
Tumor-informed.
Yeah, where you first sequence the patient's original tumor tissue to find its unique mutations. Then you design a highly sensitive test, usually based on sequencing, to look specifically for those mutations in the blood ctDNA. Table 2 references studies using these methods that can detect ctDNA down to very low levels, like 0.01%.
That sounds very sensitive.
It is. But as the review rightly emphasizes, we still need large prospective clinical trials to really nail down how best to use ctDNA for MRD monitoring in routine practice. The studies in Table 2 are promising groundwork.
Okay, prediction is another angle. Can liquid biopsy predict a patient's prognosis, how likely they are to do well or poorly? Table 3 covers this.
Yes. Table 3 summarizes biomarkers linked to prognosis. CTCs, for instance, have been looked at for a while. The table references findings, including from a meta-analysis, suggesting higher CTC counts generally mean poorer survival OS, overall survival, and PFS, progression-free survival.
Does the table mention specifics about the CTCs?
It does. Things like specific markers found on the CTCs, like CD44 positivity, being associated with worse outcomes.
What about cfDNA or ctDNA levels?
Higher levels generally correlate with worse prognosis, according to studies in Table 3, and specific alterations, like methylation patterns in genes like LINE1 or SFRP2 found in cfDNA, are also linked to poorer survival in some studies listed there.
It's not just DNA and cells in Table 3, is it? I see RNAs mentioned again.
Right, ncRNAs pop up again. Certain miRNAs and also exosomal RNAs, like specific LNC RNAs and circular RNAs, are associated with poorer outcomes in studies cited in the table.
And what about the immune cells, the PBMCs? You mentioned them earlier.
Yeah, Table 3 includes findings on PBMC analysis too. This is really interesting because it reflects the systemic immune response. Certain T-cell populations, like activated CD8 plus killer T-cells, might be linked to better survival. Others, like regulatory T-cells, TREGs, or T-cells expressing exhaustion markers like TIMID3 or PD1, might indicate a weaker immune response and poorer prognosis. The table lists several examples.
So analyzing immune cells in blood gives clues about the tumor's interaction with the immune system.
Exactly. It provides a window into that systemic immune environment.
Okay, last big application area from blood. Treatment guidance. Helping choose the right therapy or seeing if it's working. That's Table 4.
Right. This is crucial because tumors can change, especially when you treat them. They can develop resistance, so you need dynamic monitoring.
What does Table 4 show here?
It shows that changes in biomarkers like CTC counts or ctDNA levels can correlate with how well a patient is responding to therapy and their survival prospects. The table also highlights specific biomarkers that might predict response to certain treatments.
Like targeted therapies.
Yes. For example, trastuzumab targets HER2. Table 4 lists things like finding HER2 positive CTCs or specific ctDNA changes like alterations in the HER2 gene itself, or mutations in genes like PIK3CA that are associated with resistance developing.
What about immunotherapy? That's a big area now.
Definitely. For anti-PD-1 therapy, the data in Table 4 suggests that tracking ctDNA levels can help predict response and survival. Other potential predictors listed include looking for PD-L1 expression on CTCs, or determining the MSI status microsatellite instability from cfDNA, as MSI-high tumors often respond better to immunotherapy.
Does Table 4 mention immune cells again in the context of immunotherapy response?
It does. It highlights a finding about a specific subset of CD8 plus T cells in PBMCs, potentially predicting a better prognosis with anti-PD-1 treatment. Again, integrating different marker types seems key.
So looking back at Tables 1 through 4, blood-based liquid biopsy is clearly this incredibly rich source of potential information. Diagnosis, monitoring, prognosis, treatment guidance. It covers the whole journey.
It really does. But, as we've said, and as the review emphasizes, translating all this potential into routine clinical tools is definitely still a work in progress. Validation is key.
And the review doesn't just stop at blood, right? You mentioned Figure 2 showed other sources.
Correct. While blood gets the most attention, the review also covers these other non-blood biosources. Tables 5 and 6 specifically summarize research using saliva, gastric juice, urine, stool, and also fluids collected from the abdomen like peritoneal lavage fluid and ascites.
What's the general take on these non-blood sources?
They offer unique potential maybe for answering specific questions. But the review also notes a challenge. Potentially higher variability or heterogeneity in these samples compared to blood, which can make standardizing the analysis a bit trickier.
Let's briefly touch on them. Table 5 covers saliva, gastric juice, urine, stool. Saliva sounds easy to collect.
Very easy, minimally invasive. Table 5 lists potential markers like RNAs, proteins, even microbiome changes found in saliva. But contamination can be an issue, affecting quality control.
Gastric juice collected right from the stomach.
Yeah, so it gives a very local snapshot. Because of the acidity, the review notes that ncRNAs and proteins might actually be more stable there than DNA. And Table 5 shows examples, specific miRNAs, ncRNAs, studied in gastric juice that showed pretty good diagnostic performance.
Urine and stool, also in Table 5. Super easy collection.
Absolutely non-invasive. The table mentions studies looking at urinary miRNA panels or changes in the gut microbiome found in stool, showing some diagnostic value. But again, variability is a factor. Things like hydration levels or diet could influence results. making it harder to be sure markers are truly cancer-specific.
Okay, then Table 6 focuses on peritoneal fluid lavage and ascites. Why is that important?
This is really critical for gastric cancer because spread to the peritoneum, the lining of the abdomen is common and indicates very poor prognosis. Traditional methods, like looking for cancer cells in ascites fluid under a microscope, cytology, often miss cases. Low sensitivity.
And Table 6 shows liquid biopsy techniques can improve this.
Yes, significantly. The table lists examples like detecting CTCs in these fluids, finding specific methylated DNA markers, or certain exosomal mRNAs, all of which have been linked to detecting or predicting peritoneal spread or recurrence more accurately than cytology alone.
And Table 6 also mentions some pretty advanced techniques being used on ascites fluid now.
That's right. More recent studies, highlighted in the table, are using things like multiomics and single-cell sequencing on cells found in ascites. This lets researchers get incredibly detailed information about the tumor cells and immune cells in that environment, mapping out genomic changes, understanding the immune landscape. It's giving much deeper insights into how peritoneal metastasis actually works.
Fascinating. So these non-blood sources in Tables 5 and 6, while maybe less developed than blood, have their own specific niches and potential.
Definitely. Especially for localized information or specific types of spread like peritoneal metastasis.
So we've seen all this promise really well laid out in the review and its tables, but let's be realistic. What are the big hurdles still facing liquid biopsy for gastric cancer?
The review is clear about the challenges. Improving sensitivity and specificity is still paramount, especially for catching cancer super early or reliably detecting that minimal residual disease.
Getting better at finding those tiny signals.
Exactly. And dealing with tumor heterogeneity, the fact that tumors are complex and varied is still tough, especially when you might only have tiny amounts of ctDNA or CTCs to work with. Multi-omics, looking at different markers together, might help, but it's complex.
And standardization, making sure tests give the same results everywhere.
That's a huge one. We absolutely need standardized methods for collecting samples, processing them, analyzing the data, so that results from different labs are actually comparable and reliable. That's crucial for clinical adoption.
What about the future? Where does the review see this heading?
It points towards combining different types of biomarkers, that multiomics idea again, and integrating liquid biopsy data with other clinical information like imaging, patient history, maybe even standard lab tests to get the best overall picture and improve accuracy.
Makes sense, not just relying on one piece of data.
Right. And the bottom line, repeated throughout, is the need for validation. Large-scale clinical trials are essential to prove that these liquid biopsy approaches truly benefit patients before they become standard care.
Okay, let's take a step back. We've talked a lot about the review. What's the specific contribution of this paper? Why is it valuable in itself?
Well, what really sets this review apart, as we touched on at the start, is its breadth. Its comprehensive nature, which is really well supported by those figures and tables we discussed.
Unlike others that might be more focused.
Exactly. Many reviews might look just at cpDNA in blood, or just at CDCs, for example. This one casts a much wider net. It covers the diverse biosources, blood, saliva, gastric juice, ascites, as shown in Figure 2 and detailed in Tables 5 and 6. And it covers the whole spectrum of component cells, CFD and ANIC DNA, many different types of non-coding RNAs, exosomes, proteins, immune cells.
So it really maps the entire landscape at the time it was written, anchored by the data summarized in Tables 1 through 6.
Precisely. That broad perspective makes it a foundational summary. So if future researchers want to dive deep into a specific niche covered here, say, they want to explore exosomal mRNAs from gastric juice for early detection, or validate a urine biomarker panel, or refine methods for analyzing immune cells in ascites.
They'd likely start by referencing this paper.
Very likely, yes. Because this review brought together the existing studies across all these different avenues, highlighted their potential based on the data in the tables, and provided that comprehensive baseline. It essentially says, here's the wide world of GC liquid biopsy research as we know it now. Here are the promising leads across different fluids and markers. It lays the groundwork for more focused future work.
Got it. A really useful starting point and summary for the field. Okay, let's start to wrap this up. We've journeyed through the world of liquid biopsy for gastric cancer, using this comprehensive review and its data tables as our map. It's clear this technology has enormous potential. It could really shift how we manage gastric cancer, offering a less invasive, more dynamic way to understand the disease in real time.
It's definitely a field buzzing with activity and advancing quickly. There are still hurdles, as we discussed, sensitivity, standardization, validation,
Yeah.
but the promise for bringing truly personalized medicine to patients with gastric cancer is undeniable. Research is pushing forward hard.
So let's leave our listeners with a final thought, drawing from the review's implications. We know cancer is dynamic, constantly changing, and we know tissue biopsies are just static snapshots.
Hey,
If we reach a point where liquid biopsies give us widespread access to comprehensive, real-time molecular information, how might that fundamentally change not just which treatments we pick, but the entire process of making treatment decisions and tracking effectiveness day by day, week by week?
that's a powerful question to ponder. It really shifts the paradigm from reactive adjustments based on late-stage information to potentially proactive, continuously guided therapy. A very different future for cancer care.
Definitely something to think about.